So, what we're going to be talking about today, start talking about, is different aspects related to the idea, the concept of infection. Okay. I want to highlight a few things that I'm going to put on the board. I talk about microbiome. It's a pretty interesting part of the lecture. I strongly suggest you watch it. It's really cool. I should watch it. Things to remember. Microbial communities in different parts of the body are different. Your skin microbiome is different from your intestinal microbiome. Microbial communities in the same body parts between different people are similar. Does that make sense? So our intestinal microbiomes are similar, not identical, of course, but very similar. The composition of microbiome is usually changed in the disease. Does that make sense? Now, I'm really careful here. Many people say changes to microbiome cause disease. I wouldn't be sorry. We don't really know, so change. Clear? Another thing that's entirely in the lecture notes, it's memorization thing, just to remind you. You must know sterile body sites and body sites with microbiome. So with microbiome, it covers, you must know that. So body sites with resident microbiome, okay? And actually, let's introduce the first concept. So microorganisms, they can be resident. Okay? Meaning that they normally inhabit a particular body site. Am I clear? So skin, basically the entire surface, including surface of your eyes, okay? That whole thing. Digestive tract. With the exception of esophagus, which is kind of not really colonized, everything else is colonized. Am I clear? Mouth, pharynx, stomach, yes. Oh. Probably because it's my hunch would be, if you think about kind of the dynamic that happens in esophagus, it is covered with stratified squamous epithelium, and when we swallow, food constantly slows it up. So it is possible that there's just not enough influx of microbes to colonize it on the permanent basis. Again, when we say colonization, I want to have it. When we say colonization, we mean permanent inhabitants. If you would look at any given moment, are you going to find microorganisms in the saliva that goes through the esophagus that you swallow? Yes. Can we talk about esophageal microbiome? I haven't seen anything like it. Okay? Stomach is pretty poor, but it has some stable colonization. Intestine is poorly colonized. It has some stable, small intestine, stable colonization. It kind of flourishes in large intestine. Okay? Now, respiratory tract. Above the larynx. Okay? So, nasal cavity, pharynx. When it gets to trachea, bronchi, lungs, again, we do not have resident microbiome. Does that make sense? Okay. So we've got skin, digestive, upper respiratory, reproductive system. Specifically, vaginal mucosa and mucosa of the glans pubis. Not much, but whatever we have. So don't think that like the entire vagina, uterus, and fallopian tubes. No, that uterus and fallopian tube, that stuff's sterile. Vaginal mucosa is colonized. Penile mucosa is colonized. What about urethra? Very close to urethral orifice. So like basically just getting into the urethral orifice. There's some microbes only. Urethra, ureters, urinary bladder, kidneys are all sterile. So there's four things: digestive, upper respiratory, skin, reproductive. That's colon. Everything else is very speculative. You may people ask, what about question mark next to cardiovascular system? What about question mark next to the brain? There are some studies suggesting that maybe there is some colonization, but they are very, very inconclusive. Okay, that makes sense? Now, kind of not different from resident but transient microorganisms. They can be pathogenic, they can be non-pathogenic, okay? When you have strep throat, you have transient colonization of your throat with streptococci. Does that make sense? They will go away. Clear? Good? Okay. Now, let's define what is infection. Infection equals replication of microorganisms. When there is no replication, it is not an infection. Am I clear? Infections can be, I'm going to classify them using various parameters. So one parameter that we can use to define infection is contagiousness. Sorry, let's scrape contagious for a second. We're going to put communicable and put a question mark at the end. So is infection communicable? It can be communicable or non-communicable. So examples of communicable infections are quite obvious, right? Flu, coronavirus, HIV, bacterial pneumonia, anything that a person can give to another person. Clear? And remember, all the terminology is not very, but somewhat fuzzy. So let's give examples of HIV, flu, etc. Non-communicable infection, the one that you cannot give to another person. Example would be something like bacterial peritonitis, okay? Or amoebic meningoencephalitis. Remember we talked about brain-eating amoeba? Remember? Once it's there, it's it. Okay. One of examples of that kind of gray area thing is rabies. Okay, rabies is transmitted to humans from animals. Can a human give another human rabies by biting? There are no cases like that. We don't know. We can't do an experiment, right? We can't take a person and say, you read it, you're going to die, by the way, bite that person, and that person is going to die as well. So we'll get back to rabies example in a short while. Now, for communicable diseases, there is a parameter that is called R0 for basic reproduction number. That describes contagiousness. So what it is? A few things to understand about basic reproduction number. And you may have heard some flack about it in relation to coronavirus. Basic reproduction number is a theoretical, sort of modeled parameter. Does that make sense? You understand what I mean by model? We can calculate case fatality ratio, for instance, for disease. We can say we had 1,000 people infected, 10 died. Case fatality ratio is 1%. 1% of all infected people die. Am I clear? something that you can obviously like observe. Basic reproduction number is the result of mathematical model that suggests that people behave themselves in a certain way, that people interact with a certain number of people in a given amount of time. Like it is a model number. That makes sense? So what it represents, it basically represents if you have an infected person, to how many people that uninfected person will transmit the disease. So in this case, R naught is 3. Does that make sense? Now, why this is kind of a useless parameter. Imagine that tomorrow Lakeland starts classes at full capacity with no masks. Transmission will go rampant. Does that make sense? We are at limited capacity with masks. We are careful, we're distant. We already curbed the transmission of the virus. Does that make sense to you? So in actuality, it's really hard to estimate R naught, because in most cases, once infection starts to spread, we take steps to mitigate the spread. You understand what I'm saying? Like we try not to let it spread. Got it? Now, another important thing to take note. It does not reflect how severe the disease is. It only reflects how well it is transmitted. R naught for Ebola virus is about 2. R naught for measles is 18. Measles is, as far as we know, the most contagious disease in the world. And I will take measles over Ebola every day of the week. Make sense? Because Ebola is, if I get, it's much worse. Okay? Source of the disease. So let's talk about the source. Source of infection. So, infection can be community-acquired. Coronavirus is the community-acquired infection. Flu is the community-acquired infection. HIV is the community-acquired infection. You acquire it because you live among other people. That makes sense? It can be hospital-acquired. Okay. And I break them down into 2. So hospital-acquired can be divided into nosocomial and iatrogenic. So what's the difference? Nosocomial infection here, it's the infection that you contract because you are in the hospital. Okay, so imagine that you are going into a hospital in the midst of, I don't know, initial wave of pandemic for a necessary surgical procedure, and you contract coronavirus in the hospital. This will be a case of hospital-acquired coronavirus, nosocomial infection. You go there for something else. That makes sense? Like urinary tract infections that people get in the hospital. You understand? Like they go for something completely different. They contract nosocomial infections simply because they're in the hospital. Iatrogenic infection is the result of a medical procedure. For example, somebody receives bone marrow transplant that contains latent Epstein-Barr virus. Epstein-Barr virus reactivates an immunosuppressive patient and kills him. There was a famous case when a guy died in a motorcycle crash, and his organs were used for transplants in, I think, 7 other patients. Turned out that guy was infected with rabies, he just didn't know. Like he was not yet clinically obvious. And those 7 people got organs with rabies and all died of rabies. It's a pretty terrible story. Okay? But the point is, that was a classical example of iatrogenic infection. Does that make sense? Good? Okay. Basic reproduction number. I'm trying to think what I forgot. I have a feeling that I forgot something that I can't remember. Okay. What not? Pathogenicity versus virulence. So, just doesn't go ahead. Oh, thanks for reminding. I forgot zoonotic. Yes. Thanks a million. Zoonotic infection. From anyone. Now, can one infectious agent cause zoonotic diseases, community-acquired diseases, and nosocomial diseases? The short answer is we're living in the era of such disease. Initially, coronavirus was a zoonotic infection, most likely coming from bats. Once it got into humans, it became a community-acquired infection. And in some cases, when people contract it in the hospital, it is nosocomial infection, a hospital-acquired. Does that make sense? And I can give you probably a dozen of other examples, some of them bacterial, some of them viral, that can Ebola is initially zoonotic and then community-acquired. Okay, and it was nosocomial in the outbreak of 2014. Okay, so yeah, sure, absolutely no problem. Thank you very much for reminding me of that. Okay, pathogenicity versus virulence. Just to demonstrate to how uncertain the terminology is, in many cases, nosocomial and hospital-acquired are used interchangeably. So some people, when they say hospital-acquired infection, they specifically refer not to iatrogenic transmission, not to medical procedure, but to just being there. Okay, so it kind of creates some confusion. Strictly speaking, although again, many people use pathogenicity and virulence interchangeably. If we want to be pedantic, pathogenicity answers the question, does it cause the disease? Just does it cause the disease? Okay. So for instance, staph epidermidis. Does it cause the disease? The skin, your skin microbe. Like the ones on the skin. Does it cause the disease regularly? Otherwise healthy people? It does not. Plague? Yeah, it does. So we consider plague being pathogenic microorganism, staph epidermidis being non-pathogenic. Does that make sense? The virulence answers the question, how bad? Okay? So for example, there are certain, like few examples that I can give. Common cold virus. Yes, best example. Common cold coronavirus. All coronaviruses that infect you. There are 4 common cold coronaviruses. Are you following? What do they cause? What do you think? Common cold. Is it bad? Well, sniffles, cough, mild fever. Few days and you're fine. SARS-CoV-2 can cause more severe disease. So they're all pathogenic, but SARS-CoV-2 is more virulent. If you look at the data on SARS, 2003 outbreak, and MERS, 2009 outbreak, they are even more virulent. They have higher case fatality ratio, more complicated infection process and whatnot. They all are pathogenic, but some are more virulent than others. Does that make sense? And that brings us, I know that I forgot to say something, but I can't quite remember what I want to think. And that brings us to another point of discussion, which is dose that causes the infection or death. So we've got ID 50, which is median infection dose, and LDP, which is median lethal dose. Okay. Oh, I remember. Let me put a little note here. We'll address it. Okay. Don't write anything. It's for me. I'm going to explain what I need to say. So, the amount of infectious agent that you need to infect 50% of experimental group, the amount of infectious agent that you need to kill 50% of experimental group. Does that make sense? Now, I have to say, don't go hand in hand. Say the infectious dose for norovirus, which causes gastroenteritis, is stupendously low, something like 10 parts. You can drink a vial, a beaker, full of feces from a person with norovirus. It's going to contain trillions of virus particles. And you're still going to just develop gastroenteritis. You're not going to die. Does that make sense? On the other hand, the amount of virus that you need to get to die is kind of limited. Like you can't just keep it coming. Does that make sense? So, first, and probably the most important correlation, which has some restrictions. Imagine that we are talking about two closely related microorganisms, two strains of the same bacteria. And I am telling you that strain number one, to the right of that. So number one has ID15 of 100 cells. And number two is ID15 of 1,000 cells. Okay, which microorganism is more dangerous? Don't rush to the conclusion. Think and then tell. Median infectious dose for first strain is 100 cells, and for second it's 1,000 cells. Absolutely. It will be number one, because you need less cells to cause the disease. Does that make sense? Got it? Basically, less stuff to kill for the disease. Now, you can make, so what we can say is that lower IDP or LDP corresponds with a higher virulence. Does that make sense to you? Okay. Let me immediately give you an example why this comparison is not entirely correct. Infectious dose for norovirus, which causes a couple of days of gastroenteritis, is something like 10 to 100 virus particles per dot. Infectious dose of cholera is 100,000 to a million cells per dot. So infectious dose for virus is much lower. But I would take noro over cholera every day of the week. Does that make sense? You can't really compare such different microorganisms. You can't even compare often to different viruses. What you can say is if a strain, versus another strain have such distribution of infectious doses, then yeah, inside of the same species, fine. In the table that I have in the lecture notes, I have a really good example of E. coli, different strains broken down by infectious doses, and enterohemorrhagic coli has the lowest infectious dose, and it's the most virulent of them all. Does that make sense to you? Go ahead. No, with antibiotics, no. Those numbers for humans are at best educated guesses. Well, not some of them are quite correct, but in most cases, there is a pretty rigorous procedure how you can determine ID50 and LD50. You need several groups of animals, which you infect with various dilutions of the microorganism, and then you count either how many got infected in each group or how many died, and it's a stats, it's statistics. It's all very straightforward. I've done it before when I was in grad school. You just calculate, okay? I actually was talking to a class yesterday and asked, can you do it in humans? And one student said, you can. You shouldn't, but you can. If you evil genius, you can get several groups of people, infect them, and they see how many die, right? But we don't do it for obvious reasons. So those numbers that I have for human infection, most of them are just estimates based on epidemiological stuff. Okay? Now I recall what I wanted to say here. So I wanted to mention the concept of primary and opportunistic pathogen. Let's get into the last thing. So opportunistic pathogens are the ones that cause disease mostly in immunocompromised patients. Does that make sense to you? In patients who are sick. Does that make sense? Do you understand what I'm trying to say? So let's say, what is primary pathogen? What I like about 2020 is teaching micro is so easy. Coronavirus gives all examples that I need. Okay? So coronavirus, is that a primary pathogen? Yeah. If you get infected, pretty good chance, the percentage of asymptomatic infection so far is probably no more than a half. Okay? Perhaps even less. If you get infected, you most likely will develop the disease. Okay? Can it be asymptomatic? The disease, the infection, yeah, it can for virtually everything. It can be asymptomatic. But in case of corona, most likely will develop the disease. You're all young people. Your disease will probably be cough, also sense of smell, maybe some fever. I know. And if you get to an older ones, right, that becomes much harder for those people. Okay, so for me, probably going to be a lot of muscle ache, shortness of breath. I won't be able to get second floor, stuff like that. Okay, but in all cases, it's going to be disease. Opportunistic pathogens will not affect us. We are immunocompetent people. But if we end up immunocompromised, okay, then our, say, exposure, you are in the hospital, you're sick, okay, your immune system is down, you get exposed to Enterobacter erythema. In any other situation, you would just be fine. But in this case, it can cause pneumonia. Does that make sense? A lot of microorganisms from the group Enterobacteriaceae, normal enteric microbes, they would cause specifically opportunistic infection. Remember we were talking about staph epidermidis just 10 minutes ago, how we called it non-pathogenic. Well, if you take someone whose immune system is completely destroyed and deliver staph epidermidis into a wound, you're going to get wound infection, you're going to get sepsis. Because the immune system is gone. Does that make sense? And that kind of drives home another important thing. When there's no immunity, it's easier for microbes to be more virulent. We're going to talk about it next week. And really important. You will see that we're going to talk about different microbes, especially for bacterial infection. When a microorganism, especially bacteria, get into a sterile site, they cause much more severe disease. Think of it this way. You have a lot of competition in the nose or on the skin, but virtually no competition in the heart of people. So if a microbe makes it to the kidneys, that's their realm. They own it now. Does that make sense? So yeah, that's this is it. Yeah, we got everything covered. So I'll see you next week.